Okay, hi to everybody joining this live stream. We are continuing our discussion of chemistry and molecular computing, and James has something to show here.
All right. Hi, everyone. Well, when we first began to initiate this project, Stephen shared with me some of his ideas about ways in which we can take the general meta modeling principle that we have in the physics project and in the meta mathematics project. In the physics project, it's basically the physical observer. And in the meta mathematics project, it's like the human mathematician who's able to form greater abstractions in meta mathematical space. Stephen had some ideas about how we could in turn use this in subchemistry, molecular computing, and molecular biology. And put simply, I mean, it's basically the faculties of certain kinds of molecular machines to take some underlying computation, some natural process, and make something new out of it. Basically, I mean, we'll describe it in greater detail, but that was the general intuition that Stephen had. So after some discussions with Bob, it seemed to me that the ribosome was probably a good, relatively straightforward candidate for exploring this idea. Now, I'm still somewhat of a neophyte in the area of molecular biology, but hey, I mean, we have computational language. I have, chemists to discuss with, and we have a good meta modeling framework, so it shouldn't be too bad. So
let's see if I can summarize where we're going to. I mean, you're talking about, in the end, the ribosome is dealing with kind of randomly doing things, molecules and atoms and so on, but it manages to coarse grain things to the point where it is just emitting, where in the end it's emitting amino acids in response to some RNA sequence and so on. And I suppose what you might be saying is the kind of coarse graining done by the ribosome is that you can describe it in those purely symbolic terms, as opposed to having to talk about where every atom goes. Is that kind of the idea? Or
the idea is that yes, right. I mean, the idea, why don't I go through this summary? But the general idea is that the ribosome is performing some transduction from underlying nucleotide sequence generation with respect to the mRNA and the tRNA and getting a peptide sequence. So it's transducing from some underlying multi-computation to a new computation, which is the peptide sequence of amino acids that results in some translated protein.
Mm-hmm.
Right. Okay. I think I've summarized. I think most of what I wrote in the summary I've said, but just to be rather specific, I mean, the ribosomes are going to be using these codon-anticodon pairs from the messenger RNA and the transfer RNA, and I'll kind of explain what these are to translate proteins. And yes, okay.
To create proteins.
It's called protein translation, though.
I know. But it's translation from the RNA to proteins.
That's right.
Which happens to be called translation.
Yeah, right.
Okay.
So what we're going to do is we're going to begin with the nucleic acids. So we're going to think about nucleic acids for mRNA and tRNA, mRNA strands and tRNA molecules in some arbitrary cellular environment, which we're going to, for the most part, kind of ignore. And so what we're going to do is, this can be done relatively easily. We're just going to meta model the nucleic acids as nucleotide sequence strings, and those are going to be generated in some multi-computational system. Okay. And the rules in the multi-computation are going to be very simple. They're not even going to be rewrite rules. They're just going to be sequence rules. So for simplicity, what we're going to do is we're going to compute over nucleotide triplets, because that's what codons and anticodons are. So the mRNA molecules, when the ribosome docks on the mRNA strand, it's going to be, working over the mRNA 3 nucleotides at a time. Triplet is just a codon, and in the case of the tRNA, they're these free-floating molecules, but they happen to have these anticodons that are also nucleotide triplets. So that's how we're going to be computing over these. Okay, so we begin simply. We just begin by taking our building blocks. We have the mRNA codons, which are also, at this level of abstraction, they're also tRNA anticodons, and we'll discriminate between them in greater detail later. But just to visualize, this is the kind of thing that we're working with. So this is the initial setup. These are the building blocks. So now let's just start with mRNA.
Hey, wait a minute. I have a curiosity question I mean, of the 64 codons, they are in certain buckets in terms of what amino acids they make, right? But there are also certain, I mean, this is a biology question I should know the answer to. Are there any codons that are just never, I mean, there are codons that are, that represent stop codons and so on, but this is a biology question. Maybe Bob can answer this. There are codons that do not code for any amino acids, right?
That I'm not sure about. I know there's some duplication where multiple codons will code for the same amino acid, but I'm not sure if if there are any that code for none.
Okay, all right.
Just James and I were talking about this a week or two ago, and I just have not had time to go back and
well, we can actually look up in, when we do it in our fine system, why don't we say biosequence translate and then just make the tuples, make the string tuples.
Well,
maybe you already have this.
Well, I think I've seen a wheel chart where
yes, that's right.
But I think if I remember correctly, I mean there are stop codons, which in our system are basically just ones that don't have a right-hand side. But I don't remember seeing ones that were neither a stop codon nor an amino acid.
That's what I think too. I just was wondering. Okay,
and that's how it works in general, like with a Turing machine or something, right? If you don't have a right-hand side specified, then the thing just halts.
Yes.
Okay. So let's just start with the mRNA strand. So like when I say sequence rules, what I basically mean is, so you have one codon on an mRNA strand. What will the next one be? It's not a rewrite rule. It's just we're going from one codon to the next codon. Maybe there is a good rewrite rule, but this is a very simple toy way of starting to play with this. So what we're going to do is, okay, let's talk about sequence rules and the actual ribosomal docking. So in the mRNA case, these sequence rules are going to determine the mRNA nucleotide sequence that bond to some ribose backbone and form an mRNA strand. And in this case, the mRNA strand is just a path through the multi-computation. So you take some underlying multi-computation. So you take these rules, you sample a certain number of them, you start with the start codon for mRNA, which is AUG, you run it for a certain number of steps, take a certain sample of rules, and you get a path. So in this case, if we want to take this underlying multi-computation and say, okay, I want to meta model, some multi-computation that has a particular mRNA strand, take some path through this, and each path is going to have nucleotide sequence. In this case, it's AUG, blah, 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 and it's going to correspond to some molecule.
Mm-hmm.
Okay. So what do we do with all of the other tokens that aren't in this path? Well, what we can do in this meta model is we can treat those as free-floating tRNA molecules that are just buzzing around the general arbitrary cellular environment. And what we have is we have a situation in which the RNA is going to dock itself on this strand, and then at each time step, so to speak, you're going to have these tRNA molecules that are floating around.
Okay, I get where you're going. Yes.
Yeah. Okay. So we have paths, but we also have free-floating tokens, and the idea is we're going to meta model the way in which the ribosome makes use of both. And the way the RNA makes use of both is you apply a foliation to the multi-computation, and that foliation is going to include one codon for the mRNA strand and some options in terms of trying to bind these other tokens to the strand and trying to actually yield a particular amino acid. Okay, and I mean, and you have some choice in the actual path you take, right? So these are all the possible mRNA strands that you could get.
Hold on. I mean, the basic idea here is that, what you're indicating with the events here, these are not really these are not chemical reaction events. These are events that are essentially sampling another random, base, another random nucleic acid from the soup. Is that right? Because I mean,
yes.
and then the ones that are being the the program for the ribosome, the mRNA for the ribosome, is what is determining which pieces of the soup get picked up by the ribosome. In other words, which parts of the soup the ribosome chooses to observe. Is that right?
Well, because the ribosome is ultimately going to observe both, but one of the mRNA is an actual path where you have to commit to a particular mRNA strand, whereas you can have these free-floating tRNA molecules that bind to the docking site. So we haven't really explored this before, but the idea here is that you're going to take a path, but then you're also able to use the other tokens that aren't in the path, where you're basically going to bind, one of these tokens to one of these path tokens, so to speak, in order to get the amino acid. And I'll show how that works. But yes, the idea here is, look, there's plenty of biological machinery operating in the background. You have generation of DNA, you have conversion of DNA to RNA with polymerase, et cetera. And you have these tRNA molecules that are being formed and are floating around. So the events are basically meta-modeling this underlying machinery, right? Where you have, the events in some way are modeling, for the mRNA case, the computational work that was involved in actually producing each codon and sequencing them on the strand. And in the case of the tRNA, the events are just the kind of floating around, motile behavior of these in this arbitrary environment.
No, I don't understand. The ACA is going here to a CUU. I don't understand how it gets there. How does the ACA transition to something different?
Well, it doesn't transition, but this would just be each one of these, it's somewhat abstract, right? So if you take a path, you're actually considering these as part of an actual strand.
Yes.
So to speak. If they're not part of a path, these are just sequences with which, they're like traffic of the tRNA that's passing by the ribosome. So at this particular time step, it's like, I'm a ribosome, I'm on the mRNA, and this is the codon that's available to me. And they're also at this, when I foliate it like this, these two little tRNAs happen to pass by. And if it's appropriate, they can dock, and I can use both of these to get an amino acid.
Wait a minute. The amino acid you're getting is the thing that comes from the mRNA sequence. Yes?
No, it comes from, so what you have to do is you have to dock to the mRNA strand, and then there's going to be a corresponding tRNA molecule, right? And so the tRNA molecule binds to the ribosome at one of its docking stations, and the ribosome is already attached to the mRNA. So you need two keys to get the amino acid, so to speak. This is how the ribosome works. But what's interesting about the ribosome,
can we bring up a picture of this? I need
sure.
to remind myself what
yeah, sure. Let me get a picture.
Yeah, that second image you had from our second meeting, James,
right,
I think would be a good one.
Yeah, I'll get a picture. Wow, this is a crude picture, but this is the first picture I could find. Yeah. I'm gonna put it in the notebook. Okay. Just for review. Okay, you can see this. Okay, so underlying this, we have a messenger RNA strand. So that's going to be a particular path in this abstract soup, so to speak. We have the ribosome, okay, which is clasping this mRNA strand. Okay, and then we have these free-floating tRNA molecules that can
I remember how this
can also dock to this. And the point is, if you have the right match between these, then what you'll do is they'll match, and then you'll take one of these amino acids that's attached here and you'll add it to the chain. But the idea is that this seems rather deterministic, but in general, there's all kinds of these that are floating around. And you have to find good matches between these free floating ones. And the events here are just these kind of floating traffic rules, right? Where you have general behavior of these molecules in some soupy environment. And what you're trying to do is, as a ribosome, is you're finding some instance in which these nucleotide sequences are actually connected in some path, which is just shown abstractly as an actual path in the multi-computation. All the other ones are free floating. You're going to foliate it in some way where you have the tRNAs encountering the ribosome at a certain time step,
Okay.
and you're going to.
The thing I'm not understanding is starting from AUG on the right.
Yeah.
Okay. If this is just a free floatery, why aren't there 64 events coming out of that for the 64 possible new and different free floating tRNAs that can be there?
Oh, because I sample from the rules. I don't take all of them just because the graph gets too hairy.
Okay. That was the part I was missing.
Oh, okay. Yeah. So I've sampled. Yeah. Okay.
So in a sense, there's no connection between, I mean, there really isn't. What you could do is show this picture as sort of an initial Big Bang event. Because each one of those things that's out, I'm really kind of confused here. Each one of these is just an independent comes out of the soup type sequence, correct? They're not, there's no connection. There's no causal connection between the things. I mean, it's going to get confusing because the causal graph is going to show a connection between one triple high up in the graph here and a triple lower down in the graph. And I don't get that. There isn't a causal connection. These things are sort of separately coming out of the soup, aren't they?
By things, you mean the transfer RNAs?
Yes.
Yeah, they're
right.
in the soup, and there are 64 of them out there. There's 64 kinds, and one of them, I guess they just sort of bump into it. If they stick, then a binding event occurs, and you get the amide linkage formed. Then, if it doesn't bind because it's not the right,
yeah, it just moves right on.
It falls off again, fairly quickly. And it isn't until it binds with some cohesion that you then are able to go on to the next step.
Right, but I'm not understanding the multi-way graph here, or the token event graph here, because I think token event graph,
that's just coming up with a sequence for the messenger RNA, basically.
Yeah, but the thing I don't understand is, okay, every one of those events is starting from the soup bath, the tRNA bath. It does not causally connect to a previous tRNA, does it?
No, it doesn't.
So, I mean, I claim this picture is misleading because I claim that what it should look like is just every tRNA just comes from the Big Bang, rather than the tRNAs having causal, having what look like causal connections between each other. But okay, let's keep going. I get what this picture is now.
No, I'm thinking about it. It's not causal. But there's some underlying traffic behavior of this mRNA, where at a certain time step, the ribosome only has access to some sample of them, right? And I want to just show in some multi-way system that if your ribosome that has some particular mRNA path at some given time step, you're only going to have available to you a certain number of what they're saying is incoming RNA, right?
Okay.
There's only so many that are incoming, and they're behaving, this traffic is behaving in some way, has some underlying rule. If it's the case that you don't have access to all incoming RNA, it's because you have access to some sample and the traffic is behaving some rule, and that's kind of exogenous to the model, but I still think it's fair to model it as a multi-way system. But I do agree that the causal graph is unclear to me. This is a very soupy picture, but what's interesting is that you still are able to get some new computation out of it.
Okay, well, hold on. So I mean, what happens is, at any given stage, the ribosome has only some number of tRNAs floating around that are in its vicinity and able to potentially dock to it.
That's right.
And the thing jiggles around enough that all of those 64 possibilities are probably paraded past the docking site, and so that it can find out which one docks and which one does not.
Right.
Okay. All right. I get it so far. Okay.
Cool. Okay. And okay, this is the molecule for this particular choice of mRNA strand. And these are the possible these are possible paths, basically. Molecules correspond to the paths that you could take in this.
Mm-hmm.
Okay. Okay. And okay, this is just a review. And you can do it with, I mean, you can do it with this particular choice of vertex shape function. Okay. Now let's move on to the question of ribofoliation. So here I've taken a somewhat hairier example. So I've sampled like 300 of these different sequence rules, and I've generated, some multi-computation for 7 steps. So now you get something that's a little hairier. And okay, this is for the benefit of Bob and people who are still thinking about multi-computation from a somewhat fresh perspective. So the idea is that when we have multi-computation, unlike with just a normal single-way computation, we have multiple evaluation fronts happening at the same time. So it's either you have multiple rules, in this case, they're like 300, or you have rules that can be evaluated in multiple ways. So once you have these multiple paths, and these paths have individual states, you have to determine how you're going to basically slice up this multi-way system, basically what is happening at each individual time step. And so I'm just going to call the foliation when it applies to a ribosome as a ribofoliation, just for fun. Now here's just an example. It's hard to see in the middle here where the differences are, but you can certainly see at the end that there is some difference in the way in which these graphs are being organized. So this is the same graph as this one above, though it's being run for 4 rather than 7 steps. Just I think it would be a little too hairy. So what we're going to do is there are going to be 2 choices that are made. Actually, there are 3 modeling choices that are made. So first is, unless you want to run all the rules, you're going to take some subsample of all the possible sequence rules, and you're going to pick a specific ribofoliation, and you're going to pick a particular mRNA strand. So the first example that we showed was rather simple. This one's going to be a little more complicated. So we're going to take this particular path to be the mRNA strand. So what we do is, once we've picked a particular mRNA strand, then everything else that's not in that path is going to be tRNA. So once we have the tRNA and the mRNA distinguished, what we do is for each foliation slice, we ask what are the possible codon-anticodon pairings that are permissible. And what that basically is you have a given path. I'll show this one. So you have a particular path and you're asking for some foliation slice. Let's say we're just foliating entirely vertically. The possible pairings are this one with this one and this one with this one, where you're holding the mRNA constant. You have to use the mRNA, but you can also make use of non-path states being bound to the path. And eventually what you're going to do is, once these are bound, you're going to take these possible pairings, and they're going to serve as the left-hand side of some new rule. So this is where the idea of transduction comes into play, which we haven't really discussed before that much in detail, which is you have some underlying computation. And in this case, the underlying computation is quite weak and amorphous. It's soupy. The rules are not strong rewrite rules. The causal graph is not that strong, et cetera, et cetera. It's quite primordial and somewhat random. But what you're able to do is, if you're able to take a particular path in this case, as well as additional states, pair them up, and take those pairings as the left-hand side for some other rule. In this case, the right-hand side of the rule is an amino acid, then you should be able to perform what is called protein translation. So taking some particular example, we're going to use this graph. We're going to take this path. These are the possible pairings. We put that into the rule, and we get that of all the possible pairings, these ones actually have a right-hand side. And so in this case, we get histidine, serine, and glycine. And if you're going to do it in a way that respects that order, in terms of the time steps, then you would be able to yield this protein from the translation. So this is a crude example. I mean, I'm still working through exactly how to do this, but the key point here is that we did not begin with amino acids. We didn't begin with proteins. We didn't even begin with pairings. We just started out by randomly generating sequences of nucleotides. We decided that we're going to meta model a path as an mRNA sequence and all the other states as tRNA molecules with anticodons, and we're going to pair them up. We're going to convert them, or we're going to do a rule to get their amino acid, and we're going to extract a protein from this underlying soup. So that's the preliminary take on this idea of riboception.
Okay, so let me try to understand what's going on. So basically your point is, the original question is, when you've got molecules floating around, one of the things that chemistry does is it says the thing that you observe based on these molecules is the concentration of some kind of molecule. What you're saying is, here's a different thing you observe. If you're a ribosome, and there's a little bit of confusion to me here, but I mean, essentially what you're saying is, what you're doing is the chemical observation. Okay, so take the following example. I believe it's true that crabs have mostly A and T in their genome. And I believe the reason for that is that they, in their environment, they have a lower concentration of some of the amino acids. And so they tend to like to make things out of, so in other words, that's a case where, what you're otherwise doing is what you're doing mostly is you're saying, from this completely random environment of tRNAs, aka tRNAs bound to amino acids, from this completely random environment, the particular one, the particular experience that this ribosome is going to have is the one that makes this protein, where that ribosome is primed for that experience by having an mRNA that it's bound to. And so what I'm trying to understand is, that's different from, so for example, the experience of the ribosome. Let's say that you had changed the soup so that it had fewer of some particular kind of amino acid, fewer of that particular codon.
Yes, right.
Then, for example, the ribosome might take longer to synthesize that protein because it has to wait longer to get the appropriate codons to to bind to add those particular things.
It'll be the anticodon, but yeah, that's the general idea. But just to make an even more general remark, I mean, the idea in physics is we're beginning with a human observer usually, or with observer theory, we're starting to think about instruments. But with instruments, we're thinking about particular transductions by which they're able to measure particular physical quantities. And with human observers, we're just thinking about, phenomenology of humans in the universe. With metamathematics, we're thinking about either the human mathematician who's creating or obtaining abstractions from some underlying metamathematical system, or I guess maybe automated theorem proving, which is doing it in a much more granular way. But it seems as though in biology, there's what I imagine is that there's an entire stack where at each level of the stack, and we have these towers in other cases too, but in the biological case, that you're going to have different multi-computational behavior. And I don't see how you're going to be able to have some multi-computation that really begins all the way at the bottom level and is going to, we're not going to be able to take that low-level multi-computation and model anything high-level that we care about. Well, the point here is that there's some transduction being performed by this biological machinery where there's some underlying process that's happening, and they're able to, in this case, taking some underlying multi-computation, and you're actually executing some rulal translation from the computation that's there to a different symbolic computation. I mean, in this case, the peptide chain that we have here is single way because we only have one success case for each foliation.
Yeah.
But if this was richer, it could be that I actually have multiple possibilities for each foliation, and there will actually be some sort of tree-like structure that describes the different possible peptide sequences that I can get. And then, I mean, I'm still somewhat of a neophyte here, so I don't know what you do after that, but I imagine that there's a bigger fish that comes from there and a bigger fish from there and a bigger fish from there that's making use of underlying computational work performed by one's
Okay,
predecessors.
let's talk about this case that you're talking about here. So one of the questions is, at the level of which codon floating in the soup gets picked for this ribosome, it looks very multi-computational. It looks very much like you've got all these possibilities, and you are, as you say there, just simply picking a path. Okay, although I don't really quite believe in this token event graph, but I agree with you that there are a huge number of possible kind of things that can happen and codons that can bind and things like this. And any given ribosome is picking out of that soup of all possible codons floating around that could bind. It's picking out particular ones, not only particular types of codons, but even particular codons that are anticodons, whatever, tRNAs that are that are out there. There are many possibilities.
Yes.
Now, what you're saying is it the case that those many possibilities, has biology managed to essentially make a measurement in the quantum mechanical sense, or, although it isn't quantum mechanics here, that then reduces all those possibilities to just one possible sequence of amino acids? So that then, or is it the case that there can be some sort of non-determinism that's left at that stage where the thing doesn't simply decide? The strategy of biology appears to be pick a definite protein to make from, at a given moment, you're picking, at least at this level of machinery, at the level of the ribosome, you're just picking a very specific protein. Now, I think it's more complicated than that, because I think that the whole, there's the whole story of chaperone molecules. And I don't know, Bob, or anybody want to comment on how that all works?
The chaperones, I believe, are involved in folding after the protein strand as it's coming off the ribosome.
Indeed.
Yeah, but the whole process of the ribosome is almost completely deterministic based on the messenger RNA. I'm sure there are occasional missteps where the wrong transfer RNA just happens to get used and a wrong amino acid gets inserted.
Right.
There's
you're constrained by the path, right? So once you have this mRNA path, yes, there are floating tRNAs, but you're only going to be able to actually do.
How does that work in the hypervariable regions of the immune system? How does that work? Doesn't that have a mechanism?
Bob, the hypervariable region of which, Stephen?
Of immunoglobulins, of antibodies and things. There is a diversity generator that operates that puts random elements into those proteins.
Yeah, that I'm not familiar with. I don't know how that works. Be something interesting to find out.
But you see, but the whole point of that is exactly a case where what you're saying here is not what's happening. You're not generating a definite protein.
Yeah.
You're specifically trying to generate random proteins in that case, or at least somewhat random.
Mm-hmm.
So that will be an example,
I'll look into that,
James, of where you're, how do you preserve, what's happened here? In the analogy of quantum mechanics, the thing started as a multi-way system where all these possible anticodons could bind, et cetera, et cetera, et cetera. And then it was determined that there was a particular sequence. In other words, and at that point, it's sort of all classical from then on out. So I think the issue that the immune system,
but there is one thing, right? Which is exactly what I'm able to bind to this particular codon in the mRNA path. That's still going to depend on the foliation, right? So there's still not fully deterministic, because if my foliation is like diagonal as opposed to being completely horizontal, then I have different options in terms of what I'm actually matching up here.
Really? So these codons just go in, it's three are done and then the next three. Why is that? There's no correlation. You can have any sequence of codons in.
No, but hold on. What if let me get out my little marker. What if I have some foliation that's like, It's like this, right?
Well, I don't know. And I think the problem is that we're getting confused by the fact that you are showing a causal-like time sequence, which I don't believe is real, because I think that it isn't the case. I mean, if you could tell me that in the vicinity of the ribosome, I mean, I'm not really understanding how this whole, I just don't think it's causal, because I think that the ribosome just is, there's a soup that has everything in it, and it can pick something up from that.
Well, the soup has everything in it, but I imagine that at some particular time instance, it's not like every single tRNA molecule is going to be immediately available to the
No,
ribosome.
I agree, but the sequence in which they are become available is a completely exogenous thing. So
I agree. I agree. I will say what I'm not pleased with in this current token event graph setup is it's so abstract in the sense that, this particular path is a solid path that corresponds to an actual strand, whereas everything else is these are paths in the sense that they're just kind of traffic sequences that can happen, and there's really no differentiation between them besides the visual highlighting of this particular path. That doesn't please me, because they're basically meta-modeled in the exact same way, where one is a solid, actual backbone with codons attached to it, and the other is just these free-floating molecules. But the reason why I did it this way is I like the idea. Well, partly I did it out of just resourcefulness. I wasn't sure what other options I had, but I also like the idea that if you're going to transduce from one multi-computation to another, you can take particular paths, but there's no reason why you can't also take you select a path, but you're also taking additional information from other states and how they relate to that path. That's it seems that's what the ribosome does. It's not just reading off a tape, it's reading off a tape and taking some, random kind of also molecules that are docking with it. And when that works, you get an amino acid.
I think today, I mean, for the given repertoire that we have of ribosome plus messenger RNA and the tRNAs and the amino acids that bind to specific tRNAs, we get certain kinds of proteins that are made. And the multi-computational aspect, we're looking at one particular or slice, if you will, through the multi-computational space as to how things are today. But if we try to roll the clock back and see what other possibilities there might have been, then I think this sort of methodology, the multi-computation, is a tool that could be used to explore what the other possibilities could have been, and perhaps what their probabilities might have been, and things like that. Are we where we are today because things were very narrowly channeled, and it's a very probable likelihood, or is it really just a fluke that things are the way they are on this particular rock in this particular solar system?
Right. Okay, but so let me give an example of this. So what we believe about the observation of physics from sort of the arbitrary Roolead, so to speak, is that we need this computational boundedness and the belief of persistence of observers. So the question is, for the ribosome, what is its analogous brief, so to speak? In other words, if you are going to do, molecular perception, nano perception of some kind, what attributes, like the ribosome is presumably a computationally bounded thing, but what other aspects are, you're basically asking, Bob, what ribosome-like things are there? And one of the things that we're saying, when we consider a measuring device, for example, what do we believe is important about a measuring device? In general, that it takes a large number of possible inputs and produces some symbolic output, like our pressure measuring thing, where all those different configurations of molecular impacts all lead to just a single fixed pressure, fixed force on the thing that's moving. So
what are the particular characteristics of the ribosome? And one is it has to work over some messenger RNA. So it's the ribosome's not just collecting randomly, floating molecules on both ends. Because if that happened, you lose your determinism basically, and then I don't even know if you would get meaningful. Maybe you would get meaningful proteins, but I think it would be a lot harder. It would take longer, and I think it would be harder for genetics to.
The main thing a ribosome does is it takes random free-floating chemicals and assembles them into a particular chemical. Now, in normal,
hold on, but there's an underlying strand in this case. It's not what I'm saying is imagine if the mRNA had no backbone, and both mRNA and tRNA were molecules. Right. So that's one. It's not quite persistence in time, but it's like at least you have some constant. You have some constant path that you're working with. It's also working over nucleotide triplets, and I discussed this with Bob somewhat. I'm not actually sure what the significance is. Why you can't use 4, 2, 5. I'm not sure.
It depends on how many amino acids you want. I mean, I think that the question.
No. Look, you have 3 nucleotides in each codon and anticodon, and each codon-anticodon pairing that's going to correspond to 1 new amino acid in the peptide chain. But I don't see why it couldn't be 4. And then
because what's happening is right now 20 of the cases. I mean, it's that wheel thing you mentioned. There are a whole bunch of out of the 64 possible codons.
Yes,
they are divided into which one produces leucine, which one produces, I don't know, I don't know my amino acids properly.
But you could have exotic amino acids. Bob was telling me. I mean, people.
Yes, you can have exotic amino acids.
There are some unnatural ones, especially in bacteria, and they would have different transfer RNAs that would go to a specific codon. So you have a slightly different table.
You're asking a different question, which is why is it enough to have about 20 amino acids? How do we make humans out of 20 amino acids?
Mm-hmm.
Why don't we need 50 amino acids, or why can't we get by with 5 amino acids?
Right.
And the answer I'm sure is that, I mean, I believe from like this crabs example, if I'm remembering correctly, that, I mean, well, like for example, plants versus animals have different, have slightly different numbers of amino acids, if I'm remembering correctly. Is that right? If you're a pure vegan, you have to, eat some supplement because you don't get some amino acids just from plants that us animals need.
Right.
So, I mean, I think that there's this question of it's a fundamental question about life. Why does it need 20 amino acids rather than, which you could ask the same question about universal computation? Why is the simplest universal Turing machine a 2-3 Turing machine rather than a 5-7 Turing machine or whatever else? And the answer is, I suspect, I mean, this again is quite an interesting question. I mean, what is the threshold of universal construction or universal functional construction, whatever that means? As in, if you want to make a thing that walks and talks like a living system, how many building blocks does it need?
Right.
I bet the answer is much less than 20, right? It might be, I don't know how many it needs. It's an interesting question. I mean, it's somehow, I mean, again, what is the biological? I mean, could you argue for the fundamental biological significance of, like, if of the different amino acids? Like, in other words, if we didn't have blowout 3 amino acids, we don't have them, what can we not make? Would you be able to say, oh, you can't make nerve cells if you don't have those 3 amino acids? I suspect.
Bob?
There would probably be some proteins that you really couldn't make, but I suspect that there could be substitutions that could be made, and you would still get viable proteins.
Right. So look, if the function of the protein is the only thing the protein does, it has a certain shape and charge distribution, then the question is, what if that is the measured thing, the shape and charge distribution of a protein? is it the case that you can find a sequence, you just blow out some number of amino acids and say, can I find with the amino acids? I mean, that's a perfectly reasonable science question.
Mm-hmm.
That is, if you say, take only 10 amino acids, can we make proteins that have the shape and charge distribution that correspond to the, whatever it is, 30,000 human genes or whatever?
That's a good type of question, but I don't think we should take it quite verbatim, because if we had a reduced amino acid repertoire to work with, we would probably end up with still a viable biology, but somewhat different from what it is right now. I mean, to have something like chymotrypsin that would break up proteins, in digestion, we wouldn't necessarily have to make chymotrypsin itself or something very similar to it, but something that's still functional that will break those peptide bonds.
Well, I understand that. That's why I'm saying functional universality, but that's a more complicated criterion. I mean, one question is pure computation universality is a story of, can you take this actual program written in whatever language X and compile it into whatever you're dealing with? And so the first question will be, can we take, if the quotes program for biology is just these molecules floating around with certain charge distributions, et cetera, can we take that quotes program and compile it to something with few amino acids?
I think there would be some reasonable success with that.
Right. I mean, it would be interesting to know. I mean, I think we could.
Yeah.
The question is, now that we have decent protein folding, maybe, I don't know if it's good enough to do this, but that would be a reasonable question to ask. Now that we can do, reasonable protein folding predictions, and I don't know how well those work if you go far outside the domain of proteins that biology has actually selected. But assume you can, then you can use that folding technology to ask the question, if we just start enumerating, this is a question I've been curious about for 30 years. You just start folding, take proteins at random, start folding them. What kind of shapes do they make? What is the space of shapes they make?
Mm-hmm. And do it with a reduced set of amino acids.
Right. And then ask the question, could we, in principle, now it's not what we, to get there from here question, I mean, presumably, and this is where you start kind of getting into the, okay, I mean, so what would be the analogy of this with Turing machines? You could say, I'm going to have this particular Turing machine, and I'm going to try and reproduce certain programs with that Turing machine. Let's say I've got one given candidate universal Turing machine, and I'm going to pick this collection of other Turing machines. How can I emulate one with the other? Okay, so I've done that with cellular automata. You say, I've got, for example, famous case, Rule 18 emulates Rule 90 if you take blocks that are of the form 1, 0, and 0, 0. Okay, so with respect to those blocks, Rule 18 behaves like Rule 90. So you can imagine, given a Turing machine with some particular initial conditions, it's going to behave like some other Turing machine. So that's the analog here of asking the question, given, what you want is overall quotes behavior that is a particular, molecular shape within some error bounds. And you ask the question, does there exist a program, AKA an amino acid sequence, that gives that shape after folding with this, does there exist instead of 20 to the n possible proteins, you have, let's say, 10 to the n possible proteins. Can you still do it? Can you still make a thing with the right shape?
Yeah, and it wouldn't have to be the overall shape either. For a catalytic protein, you really only need the catalytic site to be quote unquote correct. And for a structural protein, then you'd probably want the overall shape and charge distribution.
Right, but I mean,
more demanding, but yeah.
But it's just like when you say this Turing machine will never be universal. Forget it, because it doesn't have blah blah blah. There are probably analogous things here with hydrophobic, hydrophilic regions and things like that.
Mm-hmm.
That if you don't have these things, you can't make a hydrophobic and then you just can't make the right thing. But I mean, I have to believe. Okay, so I mean, that's a reasonable question that one could actually try to answer. It's a good summer school project or something, to explore this. The thing I've been wanting to do for so long: the random proteins with folding and so on.
And understanding the minimal number of amino acids that you might need, which these kinds of questions could be helpful for molecular computing too. But there's a greater question here to some extent in terms of the framework, which is, let's say you have some Turing machine that's using amino acids as its building blocks, so to speak. Well, that exists in some biological tower, but you had to get those amino acids from somewhere else. And the point of this example is to show that you can consider even random generation of nucleotide sequences, and you can still take an overall multi-computation, apply a new rule to it, and get something that's slightly, biologically coarser, so to speak. You've gone from these individual, triplets of monomers to now something that it has a higher biological value. And this should go up, and this should, I imagine that this goes lower and it also goes higher in terms of the biological tower. I think that this overall, I mean, whether or not token event graphs are really the best way to model this, whether or not there's a cleaner way to do it, I'm still open to suggestions. But the basic point here is to help to motivate this general idea that in biology, you have these you have some hierarchy of biological machinery where they're passing building blocks from one level to another. And so if we have a Turing machine that's making use of amino acids because it's inherited it from something that's underlying, and it's basically what the ribosome does, from turning these codons and anticodons, their pairings into actual protein.
Okay, but the main thing I think is, we imagine that the big picture of observers is they take lots of possibilities and they reduce them into a smaller number of sort of symbolic outputs.
Yeah.
And you're observing that the ribosome is in some sense acting as a quintessential observer.
Yeah, I mean, look, this is quite hairy, and in the end, we just get this.
Yeah,
right. Because most of this is useless. Only a few of these actually are useful.
Well, okay, that's your view as a human who is used to single thread of existence computation.
No. When I say useless, what I mean is actually what I said isn't fully accurate. If you take a particular, if there's only one mRNA strand in this model and everything else is a tRNA molecule, then very few of those tRNA molecules are actually useful. If there are multiple strands, then it becomes a different story. That's something that I haven't considered here.
No, but I'm asking the question. I'm saying that I mean, the fundamental thing that's happening is that one's going from, I mean, this is a quintessential observer type situation. The issue is that I don't think the dynamics is as rich as it needs to be that interesting in the thing that the observer is observing. I don't think there's enough causal relationship here. I think there is in some other biological examples. I just think we need, more injection of molecular biology here to know some other examples. I think this is a particular case where the thing is kind of it's going from pure chaos to, the observer, it's, in other words, I think it's more interesting. This is the whole point, is whether, okay, in this case, it is like a quantum measurement. It is giving you a very definite answer at every step. It's going to say this particular amino acid was added. As you were saying, yes. The question is there a biological situation in which there is more uncertainty about what's going on, and the final, we got a result, is multiple steps away? Okay, and I suspect that any such discussion in biology will tend to be a biological mechanism that hasn't been figured out, because people are just not used to that kind of thing. I mean, they're not used to the kind of multiple threads of computation. People are used to explanations where you can essentially draw the flow chart, yes, and it is essentially the structural paradigm for science. You can just draw what it is, and that's what it's made of, so to speak.
Yeah, right.
And I think people, to have something where there is a mechanism, where there's a thing that's going on, a cell is I don't know. I'm not really completely up to speed on the current state of cellular differentiation and the extent to which the DNA is modified by, I don't know whether it's methylation or whether it's some other thing, as you go down the differentiation pathway, and as you then kind of manage to get back to stem cells and so on from a differentiated cell, all those kinds of things. I'm out of date on how that all works. But that seems like the kind of place where one might imagine in other words, I think the places that have been well described in biology are precisely cases where multi-computation isn't going to be so important, because they're cases where there is a definite this step type narrative.
If I understand what you're saying, you're suggesting that there are probably more interesting cases than this ribosomal case, where instead of performing some computation from a soup to something definite, maybe it's just from multi-computation to multi-computation, where the first one actually has something that's causally meaningful, as does the second one.
Mm-hmm.
In this case, it's just from soup to chain, which the soup is not multi-computationally really that rich.
No.
And neither is in this case the particular chain. That's the idea. Okay.
Right. So I'm saying
let's find that example.
Yeah, I know. And I think it might be in the immune system. I think there may be things in the immune system. That's my best guess of a case where essentially the measurement doesn't happen until the end. Like, for example, in this network model of the immune system, I think one's going to see something where there's, antibodies, anti-antibodies, and so on, this whole network of things where we, as observers of what's going on, don't know what's happening there. It's just some complicated, essentially multi-computational process. And the observation is, for example, do we get sick or not? Or do we get, what does it mean when we get sick? I don't know what it means. I don't know whether that is, this whole chain. I mean, look, we've got antibodies populating some shape space, for example, and we've got densities of antibodies, and we've got an intruder, an antigen, and we've got, binding to antigens and so on. We've got, the whole mechanism of T cells and all that kind of thing. And the question is it the case, for example, I don't know if this is true, but, in our typical kind of multi-computational setup, maybe the equivalent of a black hole is what happens when you get sick. That is, normally there's a dynamic equilibrium of, some small amount of antigen is coming in, you're fighting it off, you're generating antibodies and anti-antibodies and so on. It's all in some kind of dynamic equilibrium until somehow there's too high an amount of, let's say, antigen that corresponds to something like too high an energy density. And that means that in the end, your immune system can no longer, you end up with essentially a black hole. That will be, I doubt that's quite correct, but something like that might be true. And when what you're saying is in shape space, the attack on an antigen, it might be boring, and it might be the case that, you just generate at random all these antibodies, and the ones that happen to match in shape space, okay, they're winners. There's some simple mechanism where you get more of those, and you just keep filling the system up. Or it might be that it's considerably more complicated than that, and that you end up with, not something so localized in shape space. You end up with something that's a pretty big ball in shape space, and then it's an effect of. So the way you think about that is shape space, like physical space. You can imagine paths in shape space, and you can imagine generations of antibodies, anti-antibodies, etc. I don't know how long those generations take, a few days, I would suspect, per generation. And then you can ask questions about how things concentrate in shape space and things like that. And I think that's a more promising place where there is genuine, non-trivial, multi-computational causal dependence of, if you made all of these antibodies in this place in shape space, right, you will generate anti-antibodies that are in the same place in shape space, causally, so to speak.
Right.
Even though your whatever they are, hypervariable regions are, there is a mechanism whereby things are generated purely at random, but there is also a causal mechanism where things are not just being generated at random. But that might be a case in which that causal chain is not something that we are particularly aware of, nor something that biology has had a good time modeling, because it isn't a structural kind of model where you just say it does this, then this, then this. It's generating, a giant, weird geodesic ball in shape space or something. So anyway, that would be my thought about this.
That's interesting. Okay. So instead of considering the dance between codons and anticodons, it might be between antigens and antibodies in the immune system. I have to learn more about immunology. But is the point with this black hole analogy that if at some point in the multi-computation you have an antigen, maybe you have an antigen in some quantity, and there's no matching antibody or antibody support, so to speak, then the thing comes to a halt.
I'm not sure that's quite the analogy. I think the analogy, I'm not thinking about the singularity, I'm thinking about the inexorable, I don't know. I mean, I don't know whether that analogy quite works. But I think the point here, I think if we're going to look for existing biology, first question is, we've been talking about chemistry before, where the traditional view of chemistry is pure count, things by concentration, which is completely not what's going on with the ribosome. The ribosome, if there is, and I'm sure there are biological examples, we need to find that out from somebody, where, for example, you're in an environment where you're sucking amino acid X out of the environment, and therefore the ribosome is steadily trying to build a protein, and oops, it doesn't have enough. I don't know. I don't mind amino acids. Tyrosine, that one, TYR.
Yes,
that's one. It doesn't have enough of that in the environment for whatever reason. It's been leached out of the environment. Who knows why? Mm. What's the ribosome going to do? Well, in the end, the ribosome doesn't really care. It's just going to take longer. So long as there's some of that, even if it's down by a factor of 10 in concentration, as far as the ribosome is concerned, it just chugs along. It will block basically until it finds enough of that amino acid or that type of codon, and then it will just keep going.
Yes.
Right. And I guess that the question is, that's essentially a very good measurement device, because it's just saying, that's a huge reduction in, there's lots of states of the microscopic system that it just doesn't care about, because it doesn't care how many of this there is, that there is. It's just going to make this particular thing.
That's why I thought it was interesting.
Yeah, right.
In this case, again, you can see the bushiness of this, and there's just nothing, for the first two foliation slices here. There are no good matches.
Yeah. My only objection here is the sort of causalness or lack thereof of what's going on.
Well, but I guess the idea was that, look, in the background, the mRNA strand, that strand still has to be pieced together, and the floating tRNA molecules, they still have to float around in some way. There are some underlying processes that are going to dictate what the actual strand is like and what the overall background behavior is like.
Yeah, but by the way, there's a question here from SL8 live stream or comment about, if you're just picking proteins at random and you don't know what what the function is, then how can you pick the good proteins from bad? Absolutely. I think that's, in other words, one thing you can do. Let's say you're picking good Turing machines from bad.
Okay.
You can say, this Turing machine, I've got this list of 20 things I want the Turing machine, 20 other Turing machines I want this one to be able to emulate, or 20 computations I want it to be able to do. Right. Then essentially you say, can I make it do those? Okay, great, then I'm a winner. Can I make it emulate any Turing machine with sufficient effort? Then sort of I'm a winner. In the case of these proteins, if you make a protein that's some random shape, who knows what it's going to be good for? I mean, it reminds me of the story I've told a zillion times about my one afternoon of studying shell shapes at the Natural History Museum in Chicago, looking at the mollusk collection there with the curator, and trying to fill in what shells are there that are this particular shape in the shape space. And the answer kept on being, well, there is a shell. Let me go find it from this drawer in this corner. Oh, that's a shell that wedges itself in the rocks. That's a shell that is that way because it sits on the bottom of the ocean and it starts picking up things from sea anemones or something. I don't remember. But in other words, to every shell shape, there is a function.
Right.
And it's not clear, I suspect, with proteins, that we've got some random corner of the set of possible protein shapes where the proteins we have, let's say, I don't know, actin filaments or something, could that work differently? Well, maybe we could have, if we'd had a different actin, maybe we would have wheels. Actin is good for linear kind of extension and pull it in, push, it's good for pulling basically on a line. Perhaps there will be some other kind of protein, which if that one had been one in the early history of life on Earth, we would have had, I don't know what, I mean, we'd have all kinds of weird appendages that work in a different way. And, but some of those things, and I think this is just like when I've studied simple programs in the computational universe, the Fibonacci sequence is pretty common, right? That's a pretty generic thing, but periodic behavior is pretty common. Nested behavior is pretty common. There's certain kinds of things that are pretty common in the space of possible simple programs. And it could be true that in the space of things that are sort of lifelike kinds of things, that there are certain attributes that are pretty common. There's certain things, like it could be the case that, for example, electrical activity and nerve cells are somehow pretty common. It could be the case that, things that pull, like actin, for example, are pretty common. And it could be that things that, and that's a good question for biology. The fact is, biology, like technology, is surprisingly uninnovative in the sense that many inventions have been made only once. Some inventions have been made multiple times, like eyes have been made multiple times. Although I don't know how similar, that's a question I should know the answer to and don't. In crabs and things like that, I don't know whether their photoreceptors have the same, I can't believe they use, it will be rather bizarre if they use the same photosensitive protein molecules. Anybody know?
Same rhodopsin or not?
Yeah. Is it rhodopsin at all? What does a crab use to see?
Yeah, if it's rhodopsin, that I don't know.
Right. If it's rhodopsin, then I don't believe they were independently evolved. Because that's the question: is it? This is something one should know the answer to. I don't. Is it something where this is something we should look up? Is it the case that these different cases where functionally things have converged, do they molecularly work the same way? They better not, or they didn't converge, in my opinion. In other words, that's to say, it's the same question, same type of question as given air and water, they both have the same fluid dynamics. In other words, to the human observer, they both obey the same fluid equations, even though their microscopic dynamics is different. And that's analogous to saying, we've got not rhodopsin, but something completely different, but to the level that we care about, which is, can the trilobite see? The answer is yes, it can. And
in the case of trilobites, I do know that their lenses were made out of calcite, and instead of some kind of protein, the way our lenses are.
Interesting. Huh. I did not know that.
Yeah.
That's cool. That's a pretty cool mechanism. But modern insects and things which have those same ommatidia type things are not that they're I don't know what they're made out of.
Yeah, that I don't know either. But those are some very good questions there, about what's the photoreceptor molecule, and possibly also the eventual transduction to a nerve impulse back to the brain.
Yeah. Well, like for example, has something like actin been invented more than once, or is there only one actin? I've only ever heard of one, but I mean that's a question that is answerable, that somebody would know the answer to. We should ask. But I think, I mean, I mean, for our purposes, I mean that again, this is more going towards evolutionary biology and the question of sort of what is the observer for evolutionary biology, because the observer doesn't really care whether the rhodopsin molecule is made for the functionality of biology and whether the animal lives or dies in a particular environment. It probably doesn't make any difference whether the rhodopsin molecule is this or it's some completely different protein. So long as it is a photoreceptor that can see photons coming from the sun, you're good.
Yeah.
And so I think that's in a sense, that's the coarse graining, that's the observer type story of evolutionary biology, is sort of don't worry about which particular molecules it is, so long as you achieve certain functional objectives.
Oh, I see. Just like abstraction in mathematics or innovation, say in economics, the tower of evolution is basically a functionality that satisfies certain, I guess, fitness conditions.
Right. I mean, it's like the Pythagorean theorem. There are many different microscopic realizations of that, just like there are many microscopic realizations of being able to see light.
Now, we're discussing subchemistry, molecular computing, immunology, molecular biology, and evolutionary biology. Oh, and immunology all at the same time. In the case of molecular computing, maybe we can discuss how we can employ some meta model of observers at different scales in order to actually help to guide the design of molecular computing, because it's different than asking questions about evolutionary biology.
Well, I mean, this is the question of can we crib it from biology, so to speak, or do we have to invent it ourselves? In other words, can we take, I mean, many of the things that we do in computational language and elsewhere are basically sort of idealized copies of things that we humans just sort of naturally do. And you might say, well, that's kind of a wimp out. Why are you doing it that way? And the answer is because if we didn't, everything would be totally alien and it would not connect to anything we care about. So the case of molecular computing, I think the challenge is not so much have the molecules go in a big dance. The question is, can we make some aspect of that dance be something that we care about? And that's why I think that our best bet is probably the immune system, because that's a case where, I mean, there may be other cases too. I kind of feel like the two most sort of, in a sense, neuroscience and immunology are two places where they sort of have a certain degree of mystery about them. And I think that mystery might be the result of the fact that they actually involve multi-computation, which is hard to make flowcharts about and really understand what's going on in. And then the question is, for those different kinds of things, is it the case that the stuff we're measuring is just not, the expectation would be the stuff we're measuring is just not what it's not the useful thing to measure, so to speak. It's not the thing that plugs into the analog of general relativity for that kind of multi-computation. It's measuring some particular, aspect of some of something where we, and this is why sort of spooling out the protein, I don't think that's the best example, as I said, because I think it goes too directly from the soup to the result. But that's the question of what do we spool out? And in the case of multi-computation and molecular computing, I mean, the real question is, how do you get an answer? And this is the same thing as the S combinator universality question. We know it doesn't get an answer by going input, crunch, 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 output. We know it, quotes, gets an answer by its whatever course of business or something, by its sort of series of, by its dynamical process.
It's really logical behavior, right? Rather than its output.
Yeah, I'm not. Yes, it's really logical behavior, right? I mean, it's dynamics. It's what it's doing on an ongoing basis.
Yeah, right.
I mean, it's like saying, yeah, I mean, we like in a cellular automaton, if we say what really matters to the output. Okay, here's actually a good example. Okay, so imagine we are successful in making random crystals.
Okay,
and that is that we've got constraints which cause us to generate the only possible arrangement of atoms is one that looks like Rule 30, let's say. Then what could be the case is that we say, well, what matters there? What are we going to measure about that? Well, it could turn out that the triangles that exist there, which are exponential, happen to have an exponential distribution of sizes, that by golly, that distribution of triangles, we're going to use that to make a sieve. And our sieve is going to be, let's imagine, this is an amusing idea, actually. Maybe there's I need practical biology thing here. In a gel, for example, like an electrophoretic gel. Okay, so here's the idea. Let's imagine we can make a crystal that has holes of an exponential range of sizes that are just the triangles of Rule 30, for example. Does that then allow us to make a measuring instrument of a different kind than we've had before? If yes, then what we've done is to take the dynamics. Right, because those things are being formed in time. I bet that does it. I bet that's a different kind of thing than we've ever seen before. That is, if we could take one of those, or maybe, no, it's not the case if it's a crystal, but anyway, but I mean, the basic point is, what matters in the Rule 30 pattern? It could be, well, here's the output, it's one, some sequence of bits, or it could be something more related to the dynamics, like, oh, there are these triangles and there's an exponential distribution of sizes of triangles. And that's the kind of thing we're looking for. That's the kind of this is the precise input-output pair type thing. That's the kind of thing that we're looking for that generalizes the notion of input to output computation.
Yes.
And I don't know exactly where to go with that. So I mean, we're making slow progress here. We're gradually kind of understanding more. I'm thinking of doing something really terrible here, which is that, James, you had fed me last night these ideas about Eames and the hyperrulliad.
Yes.
And I would enjoy chatting about that for a few minutes.
Well, sure. Here, I can stop sharing.
Okay. Well, and
let's chat about that.
And this is okay. So everybody's welcome from our chemistry team to stick around. Although this is going to be weird, and it's going to be about a different so as people may remember, so what I realized is, so what James commented on is in the rulliad, the entangled limit of all possible computations. What is the raw material on which the rulliad operates? The raw material are Eames, E-M-E-S, which are atoms of existence. And in the case of space, those are instantiated, we can think of those as atoms of space. So the issue is, and I'm going to go, we get to actually use this again. Thank goodness for that. Hyperrulliad. It has not seen a lot of use yet. Okay, so James's observation is that the proof of uniqueness of an Eame, of an atom of existence, cannot be made in the context of the rulliad alone, but requires regression to the hyperrulliad. Okay, so
just for some context, right? So Eames have to They possess two properties. So the first is that they exist, which is important. And the second is that they have UUIDs. Okay, they have to exist because if EAMs don't exist, I mean, they're the primordial building blocks of the Roolyad. So if the EAMs don't exist, then the Roolyad doesn't exist. But they also have to be unique, because if they're not unique, you can't distinguish between them. And I would say if you can't distinguish between the EAMs, then there's no reason to say that there are multiple EAMs, and then you basically have a big crunch in the Roolyad and the whole thing will collapse. So you have to have both of them.
You know,
Yeah, go ahead.
Okay, so first point about the Roolyad is one of the key issues is, the Roolyad in its rawest state, trees out everything. Okay, but there are equivalences which make the Roolyad non-trivial. Okay, but those equivalences we have said are, but those equivalences are being made by observers. I mean, most of the equivalencing is done at the observer level. Okay, but so now what you're saying is that if you want to know, I mean, the equivalencing of EAMs and the fact that they are not all equivalent, you're saying is something that is outside of the observer. The non-equivalence of EAMs, in other words, being able to tell there are EAMs that the observer can equivalence.
Yes.
The claim would be that going the other way, that maintaining the EAMs as inequivalent is something not within the power of the observer. That is, an observer can equivalent. The claim would be an observer can equivalence EAMs,
but only because they're by default unique in the Roolyad.
Right, can equivalence EAMs for their own purposes.
Yes.
As an observer, but an observer can't establish the inequivalence of EAMs. In other words, an observer can just say, look, I believe that something definite is happening, therefore I'm conflating all these different things.
Right. But okay. The Roolyad can't even establish the
That's correct.
Of the EAMs.
Okay, so let's look at the analogy to black holes. Okay, so I claim that the termination in a space-like singularity, termination of in a at the center of a black hole, is where time stops. Right. So I think that there's a hierarchy of times. Okay, so actually, let's talk about this. So the hierarchy of time. Okay, so our usual time, a Turing machine is, a Turing machine works the way it usually does. Okay, so in the hyperroolyad, in the first level of the hyperroolyad, the level 1 hyperroolyad, a Turing machine, what would be infinite time for a Turing machine, it infinitely compresses time for a Turing machine by being able to say that, the halting problem for a Turing machine can be solved in the level 1 hyperroolyad.
Right.
Right, by having one level of oracle. Okay, but now let's go down to below our level. Below our level, time has stopped, or time is finite. So our, in black hole time, time is finite. You only have a limited amount of time. So, i.e., it's equivalent to countable numbers in this case. Okay. In this case, it's equivalent to a finite set of numbers. Whereas in the level 1 hyperrulliad, it is, I don't actually know I would guess that's somewhere up the transfinite hierarchy. Don't know exactly how that works.
That sounds right.
Yeah. So time is somewhere in the transfinite hierarchy.
I mean, is it Aleph? Is it Aleph 1?
Don't know.
For the first hyperrulliad?
I don't know. That would be the obvious possibility, and that may be related to the continuum hypothesis, whether that's actually true or not. That's a thing to just figure out.
Yeah. Right.
But then, so my basic thought would be, if you're on black hole time, then I don't quite see how this works, but I'm kind of thinking that the EAM that is at the center of a black hole, that is eventually the EAM that you just stuck on that EAM. Yeah, I mean, okay, claim would be the EAMs in a black hole, of which there are a finite number in some sense, can't determine, question mark, that they are distinct from the EAMs in another black hole. I'm not sure if that's right, but I think.
What do you mean? Because time has stopped, so there's no computation by which to.
No, I'm claiming that they have to be embedded. You know, the only way they can distinguish is by their embedding. The only form of distinction is their different position in the ordinary universe. But to understand that the fact that they have different positions in the ordinary universe is not something that can be determined, I claim, with a finite number, with time being finite in a single black hole. That isn't quite clean. That argument is not quite clean. But the thing that I was going for was, you know, the big question is, can the universe prove, aka the Rulliad, prove its existence and uniqueness? And the answer is undoubtedly no. Mm-hmm. And can it prove its completeness and consistency? So I think the consistency, that is, what would this mean? Yeah, can the Rulliad, okay, the completeness of the Rulliad is basically something about the connectedness of the Rulliad.
Yes.
Can it prove its connectedness?
Can't prove. I mean, I imagine the rule, but it depends on how you want to define consistency. But if there are rules in the Rulliad where it's the same rule, but it has different evaluation paths, well, that's inconsistency. The Rulliad's going to be wildly inconsistent because you have many rules that can be evaluated in different ways and give different outputs.
I see what you're saying. So you're saying if inconsistency is somehow, well, I don't know, it's somehow related to this equivalency thing. But I think the thing that's sort of fun about this is, just like if you're inside arithmetic, you know, inside arithmetic, you just have to assume completeness, you know, you just assume completeness and consistency. So I'm claiming.
Okay.
I mean, I don't know quite how this works, but I mean, I'm claiming that within the Rulliad, there is what amounts to an article of faith that the EAMs are inequivalent, and that is ultimately unprovable in our universe, would be the claim. And the only proof would be from looking from outside the universe, from the hyperrulliad, which we can never do.
But if it was false, I mean, what I've presumed is that if the eames aren't unique and they're somehow the same, well, then the Reuliad would collapse. So the fact that the Reuliad hasn't collapsed.
I don't think that's right. I think what's right.
Well, do you think it? I mean, imagine if eames weren't actually unique. Do you think that the Reuliad would be able to stand? I don't think it would.
No, I don't think it would. I think that if but remember, the raw Reuliad, everything is unique. It's just treeing everything out.
Well, but that's because the eames are unique, right?
Yes. Okay. So the default case, assuming eames are unique.
Yes.
We got to make eames not be a spelling word. Anyway, and Reuliad. I mean, I'm kind of upset that Reuliad hasn't been added to the spelling dictionary yet. We need to get that done. Okay, the default case, the Reuliad, I'll make some silly joke about it, but okay. The default case, assuming the eames are unique, is everything gets treed out. But then the observer, which is no good to us, then observers do trivial or quotes trivial or non-trivial equivalency.
Mm-hmm.
By trivial equivalency, I mean that the state is the same, the graph is isomorphic, those kinds of things, which seem to us like things that you couldn't fail to do. Like if the graph is isomorphic, if the graph of the universe is isomorphic, it's the same universe, we would think.
Right. But that's just because we do it by necessity, because we're computationally bounded.
Yes. But I mean, the question is, your statement is, if it wasn't, if the eames were not in fact distinct, see, I have this feeling that the only way you can tell that they're not distinct is in the hyperreuliad.
The only way that you can tell that they're not distinct. Well,
you're saying if they were the same, if they were not unique.
Yeah.
Then your claim is that you cannot support that the observer would never consistently be able to set things up with the assumption they were distinct. That assumption would eventually fail.
If they're not distinct, then they're the same, right?
Yeah.
And if they're the same, I don't see how you can build a rule. I mean, you wanted eames to have UUIDs for a particular reason. If they're all the same, I don't see why the Reuliad doesn't just collapse into a single eame.
I agree. I think it does. Yeah, right. So I mean,
right. So the point is that the point is that you have to get the UUIDs from outside the Reuliad, right? They're being imported from the hyper-Reuliad. And I think that's what hyper-eames do. So the hyper-eames are the eames of the hyper-Reuliad, and they do the undecidable computation, or they are the undecidable computation that assigns a UUID to each eame. And that makes it so. Oh yeah, yeah.
Right. But I mean, this is relevant to the existence of the universe, because, I mean, if all the eames were non-unique, then there is no possibility for existence in the universe.
That's right.
So that kind of throws you into something which I always suspected was the case, that the proof that the universe exists, in other words, that the eames are unique, that proof somehow has to live outside the universe.
Right. That's probably why there's, theology and these other kinds of fields, maybe.
Well, I mean, the point is that the argument for the existence of the universe that I've been making is, I see, the claim is that the Roolead necessarily exists. Your claim is there's an assumption required in the claim that the Roolead necessarily exists, which is that any atoms of existence, any distinct atoms of existence exist. If the atoms of existence can't be proved to be distinct, then I see. Your claim is that the formal necessity of the existence of the Roolead depends on an assumption that it is possible to have distinct things.
Yes. And in taking that further, it means that if the Roolead exists, then the hyper-Roolead also necessarily exists. See, before, the hyper-Roolead was something, at least as I understood it, where we're thinking, okay, well, what about hypercomputation? Well, let's put that in the hyper-Roolead. But now there's it seems there's a stronger statement, which is you actually need the hyper-Roolead, and the concrete support provided by the hyper-Roolead is at the level of the hyper-eames, which give uniqueness to the eames.
If things could never be distinct, there could be no Roolead.
Right.
And there could be no existence in general.
That's right.
But the fact that things can be distinct and that they can be proved to be distinct, you claim rests on this infinite tower of hyper-Rooleads.
Yeah. Yeah, because then the hyper-eames have to be distinct too. They're going to depend on some hyper-Roolead, et cetera, et cetera.
Okay, so the possibility of a distinctness then depends on infinite hierarchy of hyper-Rooleads. This is so frigging theological. It's crazy. I mean, I bet that there's actual arguments from 500,000 years ago or something about exactly these types of things, about what do you get if, it's either that or mathematical logic. Take your pick. It's either the theologians or the mathematical logic people who might have thought about this and come up with something reasonable. I'm kind of going to bet on the theologians between those two. Sorry to mathematical logic people here.
Well, I remember you gave this talk a few weeks ago, and the title of the talk was "One Roolead to Rule Them All."
I didn't make up that title. Yeah,
okay,
right.
That was the title, right? But I think the actual title of the conference was something like "Ground Truth" or "Turtles All the Way Down." And so it seems as though there's one Roolead. I don't think it rules them all, and it might be just hyper-Rooleal turtles all the way down.
Yes, it's on the back of an infinite number of hyper-Rooleal turtles. Yeah. No, it is interesting. The concept that the possibility of existence depends on things that we are permanently unable to, concretely. Yeah, I mean, it basically, where we can never know. That's interesting. I mean, in other words, humph. Well, this is it puts another twist on the whole "why does the universe exist" question. As I say, because you're saying that the existence of the Roolead depends on an assumption, and that assumption can only be justified outside of the Roolead.
Yeah, I mean, it's almost an axiom. It's like an axiom of uniqueness for eames.
It is. It's an article of faith about the Roolead, basically. If you assume that, you're basically assuming two facts. One is a contingent fact. We are in the Roolead rather than hyper-Roolead. That is where we happen to, where we as observers happen to hang out. just as we happen to hang out in this particular galaxy. And that's point one. And point two, you're adding the additional idea that that it has to be possible to make a distinction between things.
Yes. I don't even know if it's an assumption. It's just a structural requirement in order to have the Roolead. But that structural
I
requirement
know,
can't
but
be
it
supported
might
within the
might be
Roolead.
right, but it might be the case that that structural requirement is. I mean, how could that fail? Because I would argue that the same argument that leads to the Roolead also leads to all the hyperrooleads. That is the same argument of formal necessity leads to all the hyperrooleads. But your point then is that fine, that's all well and good. I see. So the main point here is that there's a shadow of the infinite tower of hyperrooleads that is a shadow into the Roolead. That is what causes the Roolead not to collapse. So okay, so the claim would be the non-collapse of the Roolead is a shadow of the tower of hyperrooleads.
Yep.
And the analogy of that for a black hole, I'm not quite sure about, but I bet there's an analogy, and I'm really late for something else.
One other, not to induce vertigo, but one other provocation. If we were observers in the hyperroolead, would we just think that we're in the Roolead and that the next hyperroolead down was actually just the hyperroolead?
Okay, that's a good question. If we were, in fact, in the hyperroolead, would we know it?
And if you don't, then what it means is we have absolutely no idea where in the tower we are.
Let's think about that for a second. I mean, we pretty much know we're not, at the base level, in a black hole where time stops. We know we're not in a thing where time stops. Because, Look, here's the point. The point is, observers like us believe we persist in time, and we have a decent amount of time for which we persist. That is, we don't just persist for
we're not Boltzmann brains, right?
Yeah,
we're not ephemeral,
right. And okay, so we believe we persist for a decent amount of time, but in the hyperroolead, I think the analogy would be that we believe we persist for a certain amount of transfinite time. And the question is, can we tell the difference between persisting within a hyperroolead where everything is operating in transfinite time?
Right.
Can we tell the difference between that and the case where we are in countable time, so to speak?
Yeah, right. I mean, if we're just computationally bounded and we're in the Roolead, or if we're hyper computationally bounded and we're in the hyperroolead, would that feel any different to an observer?
I bet it does. Because I bet it does around black holes. I bet that there is insofar as black holes are a base, but I bet we may not even know the answer yet between those two.
What do you mean by base, by the way?
Well, I claim that zero, which is, in the end, time in a black hole just stops.
Are you saying that time having stopped in the black hole, is that something that you're saying is kind of threaded across these different Rooleads, like it's common to them or something?
No,
like
I think that to the hyperroolead, they will be saying, look at that black hole in the hyperroolead. It's just a Roolead where time which we can't see into because it has an event horizon,
Mm-hmm.
but where those poor schmoes in the Rouliad, they are living only in non-transfinite time. We say those poor schmoes in the black hole, they have only a finite lifetime. They're going to just stop.
Yes.
And to the hyper-Roulians, so to speak, the pure Roulians.
We're like a black,
we have this. Our experience of time is, those poor Roulians there. They can't tell that this Turing machine is going to halt or something, or they can't. Yeah. Anyway, more to think about here. I'm sorry. This was sort of a distraction, but you never can. If we didn't have
it's good to celebrate the fact that the Rouliad hasn't collapsed. Taking some time to celebrate that seems worthwhile.
and that we're not right. That we are. Well, we believe that. Yes, we believe we have. See, we think we're all good. We have an infinite time to figure everything out in our universe. But to the hyper-Roulians, they're like, what do you mean you have an infinite time to figure things out? We can figure out, we have oracles for all our Turing machines. So your infinite time is going to get you basically nowhere relative to where you can get to in hyper time, so to speak.
I know, but then the hyper observers have the same.
I know it's all a chain of
of disdain. Yeah, right.
But what about going the other way? Is there a sub-Rouliad?
No, because that's what black holes are.
Okay. And then one question, maybe my physics is really, really rusty, but as you approach the speed of light, time slows down. So if time has stopped, you're going as fast as the speed of light or faster?
No, it's what happens is near a black hole, the light cones tip over. So you're basically at a state. What in our models, it's pretty easy to understand what happens at the center of a black hole or a space-like singularity, which is that the rewrite rules just don't apply anymore.
Okay.
So that's it. Time is over at that point, because time is the progressive, the fact that we are having a discussion and having the experiences we're having is a reflection of the fact that we are continuing to compute.
Yeah. Yeah. So if time stops, you're no longer evaluating rules.
Right. So your computer, for example, just says, if you shut your computer down or then as far as it's concerned, time has stopped. So this is the point is that a hyper time observer, has disdain for the pure time observers in our universe. That is, and imagine all the science you can do if you can solve every halting problem. And that's the case. Imagine the level of science if you can solve every halting problem, because that means there is no computational irreducibility it's very amusing. I get a spam call and its caller ID is Oracle. I think
it's a disdainful call.
That really is quite something.
Oh man.
I mean, maybe it's an Oracle salesperson, although calling random numbers, because that's or alternatively, it's precisely this. We finally cracked it, and yeah. Anyway, if you could solve every halting problem, which means, i.e., there is no computational irreducibility.
Mm-hmm.
All right, we have to go to
I
the
have to go back. I'm going to go back to my atoms and molecules.
Hey, but thanks for the feedback about the ribosome example. I mean, I wanted to just find the easiest kind of starting point for this, but I agree that the
Yeah.
immunological examples, the evolutionary biological
this is a
examples,
good this was a good
these are
exercise.
these are richer, and so I can take what I've done here and find something
Right.
that's actually causally meaningful and do that.
Yeah.
All right, and we will continue on this more philosophical whatever discussion at a different time and see whether we can whether the hyperreal turtles are alive and well. All right, thanks for joining.